introduce Dr. Marie Gilbertson, DVM, and PhD. She is currently a staff scientist at the University of Wisconsin at the Wisconsin Cooperative Wildlife Research Center. I heard her speak last summer. When I read her bio, I thought, oh boy, this is going to be academic. But actually, she's a very engaging speaker. She is studying how diseases, particularly CWD, move through wildlife populations naturally. She has some slides that literally had some grizzled old outdoor riders on the edge of their seats. So I will introduce Marie.
So yes, thank you so much for having me. As Laurel said, I am a staff scientist at the Wisconsin Cooperative Wildlife Research Unit, so I am an employee of the university, but I have the great pleasure of collaborating very closely with folks at the DNR in doing this work, looking into movement ecology and chronic wasting disease in Wisconsin's white-tailed deer. Haha. So I like to start my talks about CWD and deer by talking about kind of why do we care about deer. Now, I suspect there's a lot of deer hunters in the room, and so I know you all care about deer for the deer hunting component, but of course, even the deer hunting component has a lot of different elements to it, right? It provides some really important conservation funding for a wide variety of species and research funding as well. Deer hunting is also a really important part of cultural identity, both for the kind of traditional sense of the Wisconsin hunting, but then also the sharing of meat with elders can be really important in some indigenous communities. A healthy deer population is also an important part of a balanced ecosystem. You know, they're important herbivores and also an important prey species. Too many deer or too few deer can be a problem. Deer can also be a really important avenue for kind of the general public to interact with nature. I'm a suburban kid growing up, and you know, deer were like the biggest wildlife that you would see in the kind of parks and stuff around you. And so they can be a really important way for people to feel connected to nature. And so I like to kind of sort of start from that point of why we kind of really care about deer, and that it's for a multitude of reasons in terms of supporting both human culture and identity as well as healthy ecosystems. Of course, here in Wisconsin and across much of North America, chronic wasting disease, or CWD, is this really important disease of our deer species. It is a fatal prion disease of cervids, so it's been found in whitetails, but also mule deer, elk, and caribou. And so it's a really important disease here. It's always fatal, and it's a prion disease, so it's related to things like scrapie in sheep or mad cow disease that you see in cattle. CWD is transmitted both directly between deer, but also through the environment. So if a deer defecates into the environment while they're infectious, those prions can survive for potentially years in the soil and cause new infections in other deer. CWD has a very long incubation period. So the time between when a deer becomes infected and when they first start to show clinical signs can be years even. And it is generally years before they end up dying of their infection. When they do start to show clinical signs, those are things like wasting, where the disease gets its name. So they lose a ton of weight. But you also see things like lethargy, so they don't have very much energy. You'll see polydipsia, which is our fancy veterinary word for increased thirst. And we also see ataxia, or uncoordinated movements, because it has these neurological effects as well. Once you start seeing those clinical signs, death is typically within weeks to months after that point. In terms of transmission, however, which is a really important part of how we potentially manage this disease, we have a ton of open questions about what is really driving or shaping transmission of CWD. We suspect that where animals spend time is really important for transmission. If you've got resources that are very attractive, that can draw in different social groups of deer, that can potentially foster transmission events. We also think about things like long distance movements, whether that's dispersal or migration if you live, say, in northern Minnesota. In those cases, deer making those long distance movements can be transporting prions into new areas or bringing CWD into new places. There's also lots of open questions about the importance of deer carcasses. So if a CWD-infected deer dies on the landscape, they create this hotspot, this sort of bomb has gone off of prions into the soil that can potentially last for years and foster more transmission. And so there's, again, a lot of questions about how long do those prions last? Does it matter what the soil is like there? And also, does it matter what kinds of predators and scavengers might be involved? What's the role of humans in this whole thing in terms of moving deer around the landscape or how we interact with deer? So again, tons and tons of questions about really what drives transmission and spread of CWD. And so that is part of where the Southwest study comes in. So the Wisconsin DNR in 2017 undertook the very large, very impressive Southwest CWD Deer and Predator Project. So if you can see, maybe if you're more in the front of the room, you can see a map of the study area here. We've got Madison over here, just off the map on the east side. Down here is Dodgeville. And so you can see kind of the extent, the Wisconsin River forming kind of the northern border, and Highway 18 down here making the southern border. And you got the little sort of inset map where you can see it up there. So in this study area, the DNR went and captured over 1,200 animals, which I have to say is just astonishing. From 2017 to 2020, 1,200 animals that they captured. That included bobcats, coyotes, fawns, and over 800 adult deer, which again is like an incredible amount of effort for any wildlife study. This involved nearly 400 participating landowners because most of this area is private land, and so have to get permission and have willing collaborating landowners. In addition, about 1,000 volunteers participated in this, especially for those detecting of fawns. And then with those deer that had those GPS collars, those collars send out a mortality signal when a deer has been stationary for a certain amount of time. So the DNR would send out then field teams who would go investigate those mortalities. So 433 deer mortality investigations were completed. Now sometimes that's going out and just finding not very many remains. You've got maybe an approximate date of death, but sometimes you've got a carcass in really good condition, and they can actually go and bring that carcass back to Madison, to the vet school, where they can do a full lab necropsy, which is an animal autopsy. So a huge wealth of information coming out of the Southwest study. Now I came in first as a postdoc, now as a scientist, in 2001. And they said, hey Marie, we've got this data, all this deer movement data, and we've got some questions, and we'd like to work with you to work on some of those questions. So I have spent the time since 2021 working with that movement data to learn what we can learn about what all that deer movement, how that interacts with CWD. So I'm going to talk about two kind of main topics in my research, or things that we've been finding. And the first is to talk about land use and chronic wasting disease. So I'll bring this figure back up because land use can be really important for shaping animal movement. Again, there might be particular habitat types that are particularly attractive during certain times of the year. Let's say you are driving along Highway 18 in the winter and you're looking out the window and you might see a big old group of deer out in a harvested cornfield, right? I think we've all had that experience driving around Wisconsin. So there are some habitats that may be kind of attractive or really fun places, or places that deer like to hang out during certain times of year. And that may mean that those are good places for transmission to occur, that you're bringing all these deer together, they're interacting, or they're sharing the same space, and maybe transmitting disease. So we wanted to learn what's going on with deer movement and land use and the implications of that for chronic wasting disease transmission. So I'm going to talk about some of this work in the context of kind of a year in the life of deer in Southwest Wisconsin. So we start off up here in the top left with our fawning season, which runs from the beginning of May to the end of June. During this time of year, we see a lot of avoidance between deer groups. So in terms of the social environment, especially does are really avoiding each other. They kind of isolate, go have their fawns, hang out, and kind of keep away from each other. They tend to have very small ranges, and they really strongly favor forest cover. Makes sense. You've got fawns, you're trying to keep them safe. You really like that forest cover. It's a good place for food during this time of year. This is also a really important time of year for dispersal. And so we did a study of dispersal in the Southwest project. So this is all of the dispersal events that we detected in the Southwest study area with all of this data. So the kind of butt of the arrows is where deer started, and then the point is where they ended up, just a straight line. So you're not seeing all of the wandering that they do in between. This map is colored by land use, and so the yellow is going to be your agricultural land, and the green is going to be your forested land. So you can see there's quite a lot of forest up in the main study area, and it's much more agricultural down here. Here you've got Madison. I suppose we're like somewhere around roughly there right now. So I want to zoom in on this particular deer here for a second, because I think he's pretty exemplative of some of the things that we were finding in this work. So you can see he's starting out up here. He's in this green forested area, kind of just hanging out, doing his thing. This is before he's really started to disperse. And then you'll see him kind of explore a little bit, and then he's going to start off and go on his little dispersal. So he's going to come down here south of Highway 18, and I want you to watch, and maybe if you've got really good laser beam eyes or something, you'll be able to see how he tends to sort of hop from forest patch to forest patch as he comes south of Highway 18. And he ends up traveling quite a long distance compared to some of these other dispersal events. And that is consistent or representative of one of our major findings here, which is that when deer are dispersing, they tend to avoid agricultural land use, and they tend to disperse longer distances when they're going through areas with a lot of agriculture. That those open fields are not great habitat for where deer want to settle down. They're not great spaces if you are traveling through a new area and you're worried about getting eaten or something like that. And so that land use is really important for shaping some of these dispersal events. But of course, I'm here talking about CWD, right? What does this mean for CWD? Well, let's say we drop CWD into a deer here versus up here. Where do you think it's going to travel faster? You know, what zone do you think it's going to be more likely to travel faster? Well, we might expect that if you drop CWD in a heavily agricultural area, those longer, more frequent dispersals are potentially going to allow that CWD to spread to new areas a bit more quickly than if it were started somewhere in a heavily forested habitat. So, you know, this part of Wisconsin versus, say, southeast Minnesota might have really different pictures in terms of where CWD might spread fastest. And that's important if you have a detection and you're setting up, say, surveillance or management zones, as you think about how big does that zone need to be. Well, we might expect deer in here are not going to go quite as far as deer out here. So it's an important thing to be considering from a management perspective. All right, so that's our fawning season, and then we shift into our post-fawning season, which really runs kind of most of the summer and then the first half of fall. There's a lot of similarities in terms of our behavior during this period, where we still see quite a bit of between-group avoidance and still have some small ranges, but our habitat selection really changed quite a lot during this period, because we saw quite a lot of attraction to agricultural and grass or pasture habitat. Now, if you've ever grown soybeans, you've probably experienced this firsthand, right? As deer are coming in and eating all the soybean crop, because it can be very attractive during this time of year. But I think it really shows up quite nicely about kind of why we care about some of this stuff if I show you this kind of case example. So we have two does here. They're not considered within the same social group, but we've got a blue one and a pink one here. And again, the background is colored based on land use, with the yellow being that kind of agricultural habitat. And you can see that really in both seasons, but especially in this post-fawning season, the area of overlap, which is outlined in black, is really centered on this area of agricultural land use. This is an attractive type of habitat, and if two deer are going to overlap, it's maybe more likely to be happening, at least in this time of year, in that agricultural habitat. So it's an important thing to think about from a transmission perspective. We're seeing between group avoidance. So these two deer might be avoiding having a direct interaction, but if they're using the same space, we're talking about an environmentally transmitted disease, so we want to know what kinds of behaviors are happening in here and what kinds of transmission is then maybe the most likely. Are these deer performing behaviors that are really likely to foster transmission events in these spaces? All right, so then we get to the breeding season. As you might expect, habitat, it's still important, but really the social environment is shaping a lot of things during the breeding season, right? We've got attraction between groups, we've got large ranges, we've got high activity. We certainly expect to see quite a lot of direct transmission of CWD during this period. There can also be quite a bit of potential for indirect transmission as well. There's a lot of research going on looking at the role of deer scrapes, for example. So those are spaces where deer are going to go and urinate, maybe defecate, and then other deer come along, they lick the licking branch that somebody else has been licking, and oh, by the way, CWD prions show up in deer saliva, right? So that can be a place to have some indirect transmission. We just don't know exactly how important a role that is at this time, but lots of research going on that. When we shift into the non-breeding season, so this is really core of winter period, we still see between group attraction. Again, picture those deer groups out on those ag fields near the highway. We see large ranges, and again, we see this attraction to agricultural or grass pasture habitat. Our deer are using larger spaces to access more resources. They're potentially hanging out with other deer that they don't normally hang out with. And so this may be a really important time for both direct and indirect transmission. And that agriculture, that land use component may be a really important piece during this time of year. So I think the breeding season tends to get a lot of attention when it comes to CWD transmission because we are thinking about the rut and all those mating events and what that means for spread of disease. But I think, it would be a mistake not to also be thinking about the importance of that winter non-breeding season and what deer are doing during that time. Again, bringing this figure back up and now expanding it out. So I showed you these two images before, and then really down here in the non-breeding season, you can see how much bigger these ranges are, how big this area of overlap is, and we're still seeing a lot of that space really focusing on that agricultural zone there. So to summarize, if you take nothing away from this section, this is the slide to pay attention to, or really just like the words right over there. So land use, sex, and season are all really important for shaping where deer are moving, that movement ecology, but then that also has really important implications for CWD transmission and ultimately what we, kind of how we respond or deal with CWD. So now I'm going to shift gears a little bit. So I've just been talking quite a lot about how movement affects transmission, affects CWD. But now I'm going to talk about the flip side of that and how CWD can affect movement and what that then means for onward transmission. Because I'm a disease ecologist and these things don't exist in isolation and you can have crazy feedbacks that go on. This is what happens sometimes with using other computers. I wondered if it was going to do this with these figures. Sometimes they turn purple. It should be these only ones. So we took our deer movement data and we found about 40 individuals who died of CWD. They died in end-stage disease, so they were very sick by the time they died. We matched those with some control deer. So they were healthy, but they were age, sex, and sort of season matched. So we could really closely match our cases versus our controls. And we looked at how movement maybe changed in our CWD cases. Now what you're seeing, maybe if you can see through the purple in these images, so we've got months prior to case death here on the x-axis. So over on the right side is going to be 1 month before death, basically. And then there's technically a dotted line right here. And if you see these down below the dotted line, that means that cases were significantly lower than controls. So what I'm showing you here is that in the last couple of months before death, CWD cases, their movement rates go down. And over here, in the last 5 to 6 months before death, their range sizes start to go down. So we see reduced movement, reduced ranging in our CWD cases in the months before they die. So we do see some changes in movement. Now that can potentially be important as you're thinking about, say, where is a deer contaminating the environment? What's the kind of spatial extent of other deer that they could potentially be transmitting to? We do care that then that movement is changing because it potentially has implications for transmission. But I want to give you a bigger picture, some bigger context. So I've got cases over here on the left. We've got controls over here on the right. This vertical dashed line is 6 months. So from here to here, that is between, that's the window that I was just showing you. That's the 6 months prior to death. What I'm showing you are all of these dots represent a deer that was captured. And on the x-axis, it's the time that it was captured relative to when it eventually died. So everybody died at time 0, and then you just see how far out it was before that when they were captured. Now, all of our cases, they're all CWD cases. They all died in end-stage disease. And part of how we defined end-stage disease was being in terrible body condition. This is body condition on the y-axis. And you can see that some of our cases, within 6 months prior to death, were in excellent body condition. But by the time they died, they were in terrible body condition. I will also say, these points are colored by their CWD status at the time of capture. So we've got some individuals who tested positive more than 500 days before they eventually died of their disease. When they test positive like this, they can be shedding at this time. They're probably shedding at this time. They are infectious. So some key things to take away from this figure. It's really likely that shedding, like these guys out here, is starting long before any changes in movement. What I showed you in terms of changes in movement, that was in this last 6-month period. But you've got guys like this who are CWD positive for maybe over a year before they maybe have any changes in their movement. In addition, we've got, again, these individuals who are in great condition within 6 months of death, but they eventually died down here in this really terrible body condition state. So it looks like condition changes very rapidly in those last few months prior to death. So we can put this out on kind of a little bit of a timeline. And so these are sort of phases that we've sort of proposed for CWD infection. So we start off with a kind of preclinical phase. A deer gets infected at time 0, and then sometime, maybe 6 months, even out to, over a year, almost 2 years, is when they start to actually shed. Now, generally, it's going to be on the shorter side of that, but it can be prolonged, or it can take longer for them to start shedding, depending on their genotype. But ultimately, they start shedding maybe 6 to 12 months after they're infected. But then it can be quite a long time before they eventually start to show kind of this subclinical phase. So we're arguing that this is a subclinical phase because if you plopped one of these deer in front of me, I, with my eyes, would probably not detect that this animal was sick. If I slapped a GPS collar on it, though, I might be able to detect some changes in its movement behavior. But to the normal person seeing that deer, it's probably going to look like a healthy deer. It's not until about 2 months prior to death where we really start to see that clinical phase. The things like, clinical wasting, hypersalivation, those kinds of changes, and then just 2 months, roughly 2 months prior to death before then they eventually die from their disease. But what does death from CWD actually look like? That's actually a question that we get fairly often. And so I'm going to talk about that. Now, I know you all just ate. I'm going to show some slightly gruesome pictures. It's okay if you look away. Word of warning. So we've got a buck here. He was captured and collared in January of 2017, and he was about 2 years old at the time. Ramalt is just a name basically for the test that we can do while a deer is alive to test for chronic wasting disease. So he was negative at the time of capture. Then, during his life, he was range resident, or at least after we put a collar on him. And then he died in January of 2018 and was found a day after death. He was in his carcass was in great condition, so they could bring him in for a full lab necropsy. He was CWD positive at the time of his necropsy. And this is about what his lungs looked like. So we've got spine up here, kind of spine side up here. His head is this direction towards the wall, and his tail is toward that wall. And this is not normal appearance for lungs. All right, so normally in a deceased animal, you're gonna see in perfect conditions, light pink fluffy lungs. Now, if they've been laying out in the cold Wisconsin winter, they might get a bit red and everything just from sitting, just kind of some of those post-mortem effects. But they definitely shouldn't be all mottled and blackened and full of nasty stuff. This guy had what a pathologist would call severe cranioventral suppurative bronchopneumonia. fancy word for saying really nasty pneumonia that's mostly kind of towards the head and the belly zone. Now, this kind of pneumonia is most commonly seen with aspiration pneumonia. And this happens to be a common sort of secondary thing that we see with end-stage chronic wasting disease. When you have clinical disease, you're hypersalivating, you have neurological problems, and we think that that is what leads to aspiration pneumonia, where an animal breathes some foreign material into their lungs and it creates a really, really nasty pneumonia. So one of the ways that CWD end-stage animals die is actually by having aspiration pneumonia. Unfortunately, they also will simply die from starvation, basically. So I'm going to show you another case here. So here we have a doe. She was captured and collared in December of 2017, approximately 3 years old. And at the time of her capture, she was test negative. She was, however, described as very skinny at capture, weighing in at about 54 kilograms. There's 2.2 pounds per kilogram, so you can roughly double that to get an idea in pounds. She died and was found on Valentine's Day of 2018, so not that long after her capture. Now, they weighed her at her time of death. Weighing at death can be a little bit like comparing apples to oranges compared to when they're alive, because she's maybe been sitting out, she might be damp, there can be some differences, maybe she's kind of dehydrated kind of thing. So it can be hard to make a direct comparison. However, at her time of death, she was CWD positive and weighed only 35.5 kilograms. Now that is a massive amount of weight loss in a short period of time. And even though there might be some sort of artifacts of, post-mortem changes kind of thing affecting that weight, that is enough that that's a real weight loss to me. That is still a massive weight loss. In her lab necropsy, the pathologist recorded that the rumen, or her stomach, contained a large amount of feed material, and she had severe emaciation with adequate feed intake. I think this is something that gets lost a lot of the time when we think about CWD or we talk about it. These animals are still eating. They can be eating an adequate amount, but they are not absorbing that nutrition and they're still losing weight in spite of eating all of that food. She was also described as having what's called serous atrophy of bone marrow fat. Again, fancy pathologist term. So if you've never looked at bone marrow, we've taken some long bones, or these are pictures of long bones that have been kind of cut and cross-section, and so you're looking at, down into the cross-section of a bone. This is the bone marrow. It should look kind of pink or yellowish, really fatty. It's full of fat. Great, rich kind of material here. This is a picture of her bones. So this is her bone marrow here and down here. And it looks like jelly, right? It looks like kind of apple-y, grape-ish, not grape, but almost raspberry jelly. And that is because she has mobilized the fat everywhere else in her body, and she has now used up even all of the fat in her bone marrow to try to stay alive. So this is something that you see in any animal that is in a severe, severe starvation state. And so this is why, end-stage CWD, this can be a really common clinical sign or a common sign on post-mortem. And why we say they do simply die from starvation, from that emaciation that goes along with severe disease. And so I want to bring it back to this figure, because again, recall that doe lost a ton of weight there in that short period between when she was captured and when she eventually died. And again, we see these individuals who are in pretty decent body condition within 6 months. So that CWD-induced wasting can happen relatively quickly in the grand sort of scope of that whole disease process. So I want to bring it back to CWD effects on movement and what all of this means here. So again, I really want to hammer home that it's likely that our deer are shedding infectious CWD prions long before they have any changes in their movement, right? We've got individuals who test positive and don't get to that point of maybe showing clinical or movement signs until much, much later, months, even a year later. So, even though this makes me feel like a terrible disease ecologist, when we are thinking about, say, using deer data to inform statistical or movement models to predict kind of where CWD is going to go next, or how do we best manage CWD, we can probably use typical deer movement to answer those kinds of questions, because for the most part, that typical deer movement is representative of the majority of the time when these deer are transmitting. In addition, I also want to raise the point that, sometimes we think like, well, maybe hunters or predators are more likely to remove CWD infected animals from the landscape because, especially like predators, they're detecting a slower moving animal or a less healthy animal. But if that change in actual like movement and health isn't until really late in the disease process, you've still got a lot of opportunity for that deer to be transmitting before they get removed sort of naturally by a predator or by a hunter. And so, I would say more research is really needed to understand how much this may or may not play a role in kind of helping manage CWD, whether through kind of hunters naturally, again, removing some of those animals or predators doing so. But I think there's, potentially a limit to just how much, say, predators can get us out of this. In addition, and this is probably the most important thing that I want anyone to take away in this room, is public communication is really, really important when we're talking about CWD. Because a healthy looking deer may be CWD positive and infectious. And you hear reports of people who say, my deer looked healthy, I'm not gonna get it tested. But I just showed you all this information about how these deer can look perfectly healthy until just a couple months before they die. So get your deer tested, tell your friends to get their deer tested, because they're gonna look healthy until they're really towards the end of that disease course. All right, so with that, I will wrap up. I have so many wonderful people to thank and acknowledge. I work with our self-named Deer Ecology and Disease, or DEAD team, which I think is the best name ever. Especially Dan Storm with the DNR, and then my supervisor, Wendy Turner, who's our unit leader at the co-op unit. I have to give a huge round of thanks to the field team, without whom none of this would be possible. And of course, massive thanks to all of our funders. And with that, I'm happy to take questions. If you would like a link to the Southwest Study webpage and you're phone savvy, you can always take a picture of that QR code to get there. So anyway, thank you. I will repeat the question. So the question was, what are the chances that the herd will kind of provide some immunity to CWD? So the weird thing with CWD is that it's a misfolded prion, or misfolded protein, right? And we don't really mount an immune response to something like chronic wasting disease. So it doesn't work quite like a virus or a bacteria where you can have this sort of natural immunity, develop a point of herd immunity in your population. Instead, it just keeps happening. We haven't been able to develop a vaccine to this because it's hard to really have much of an immune response to cause the body to sort of block this infection from happening. So really, so as part of the Southwest study, there's also been a really big kind of survival study, and they have seen that you can reach a point of prevalence at which point population growth starts to go down, that you no longer have positive population growth and your population is going to get smaller. So really, in a sense, the immunity may just be that your population starts to get smaller, so you have lower densities of deer, and that slows transmission that way, unfortunately. So we don't really have a herd immunity place to get to. Yes?
You just hit on the density question. I mean, in traditional deer management, units had overwinter goals, X number of animals per mile of deer range. And I'm not sure where the threshold is. I'm sure it's different in northern Wisconsin than it is in southern Wisconsin. Is there any drift or any conclusions or any information you can put out there in terms of what seems to be a important threshold?
So the question was about really basically, is there a threshold of deer density where CWD is maybe going to be okay? Fair?
Yeah, reasonable question, yeah. Sure.
So I love that question as a disease ecologist, because a fundamental, you know those things in your field of fundamental principles of a field, one of the fundamental principles in disease ecology is frequency versus density dependent transmission. And for a lot of diseases, we assume that as density increases, disease transmission increases. But of course, things are not as sort of clear cut as all of that. And there's literally a paper out there titled should we expect thresholds for disease? One of those core fundamental papers. So the answer to your question is, who knows? That's the disease ecologist question, or the disease ecologist reaction, I guess, to that. I would say that I feel pretty confident that CWD is a mix of kind of density versus frequency dependent transmission, which I don't know that any of you are disease ecologists, but basically that there is a strong tie between density and transmission, but that's not the only story. So reducing deer densities will help to reduce transmission to some extent, but that's going to be affected by all sorts of things like land use, demography, all of these kinds of things. So that's my still unsatisfying, but maybe better answer to your question.
Science is so messy.
It is messy. It is very messy.
More research.
Yeah, keep funding. Great question. So that question was, have we seen any CWD in kind of any of the predators or even scavengers? And so there's a ton of research that's been going on looking at that, because one of the big concerns with CWD is that some other species, including feral swine, might contract CWD through some variety of interaction with deer. There's also a concern that maybe it gets into that other species, changes a little bit, and then can transmit to humans, right? So lots of research on lots of different species. Some maybe seem like they could be hosts to CWD, but oftentimes these experiments are kind of taking CWD-infected brain material and injecting it straight into the brain of another animal, and that's not exactly a natural interaction, right? So it's hard to say exactly how likely transmission is to happen with that kind of work. You still need to kind of carry it on and look at kind of more realistic transmission events. We've also seen things like, say, a predator or scavenger feeds on a CWD-infected animal. The load of prion that goes into the animal may come out lessened when it comes out the other end of that predator or scavenger. So the predator or scavenger doesn't itself become infected, and maybe even potentially kind of sort of reduces the prion load. And so there could also be kind of a cleansing effect. So there's, again, lots of questions about exactly what role they play. There's also a question about if I, a crow, eat from an infectious carcass and then I go flying miles and miles away, pooping out prions across the landscape, is that enough to then spread CWD to new areas? So again, more research. Does it help? Yes, right here.
So you were able to detect CWD on a live animal. First of all, how did you do that? What did you use? And is there any hope of ever developing something that we can find that earlier in the deer so that after we don't have to put it in the freezer and everything, we can do it right? Anything going on there?
Great question. So the question was basically, how do you detect CWD in a living animal? And are there efforts going on to kind of detect CWD earlier or faster? So in terms of how we detect it in a live animal, so it's called ante-mortem testing, which sounds like I'm anti-death, but it's just pre-death testing. So one of the common ways that you do that is through RAMALT, which is just a long acronym for basically a rectal tissue biopsy. So there's lymphoid tissue around the rectum, and so we go and take a little butt biopsy, and we can look at the tissue and test for CWD with that tissue. There are other folks who have used like tonsil tissue. There's been efforts to use ear tissue. So there are a variety of different tissues that you can use. Now, in general, those tests are basically not as accurate as if you could take some brainstem, take those retropharyngeal lymph nodes, and do your testing that way. So they're more likely to test test negative. You're going to miss some infected deer with those ante-mortem tissue tests. As far as the kind of opportunities for faster, earlier testing, so there are some new diagnostic tests or newer diagnostic tests that folks are working very hard on getting them validated on different tissue types that are more sensitive than our traditional testing methods. I'm not sure that they're necessarily a lot quicker, but they are given like the same, if you have a deer with sort of early infection and you give the same tissue to one test versus the other test, the newer test is more likely to say that that tested positive. It's more sensitive. It's going to pick up earlier infections. As far as getting things faster, boy, that's kind of an infrastructure question, right? You need the labs, you need the people to be able to do all of that testing. And so, I think there's a limit to just how fast you can get, and I'm not a DNR employee, so I can't speak to how things go internally. But I think everybody's always trying to make it faster, certainly. Oh my gosh. All right, over here.
Do you have a way of notifying either high body temperature or lower body temperature when you're checking for apparently healthy but sick deer?
So the question was, can you check for abnormally high or low body temperature when testing sick deer and looking kind of at their condition, right? Great question. I haven't thought about that. So there's a student in our lab who actually has been working with, she studies a totally different environmentally transmitted disease, anthrax, in southern Africa. And they've put biologgers in animals to look at their body temperature and how it fluctuates over time. So there are ways that you can do that. We haven't looked at that kind of intensively. You literally have to implant a little logger inside the body, and then they run around on the landscape with that biologger. So we haven't done anything quite like that with deer, but it could be an interesting way to look at how, say, deer in poor condition are responding to winter or severe weather, and how that maybe makes things worse for them to be in that really poor condition, certainly. Did that answer your question?
Well, I was thinking you could use ultraviolet,
Oh, sure.
infrared,
sure.
To a herd and see,
Who's looking really sad? I feel like they've done some of that with mange studies too, because they lose coat, and so those animals will be losing a lot of heat if you look at them from an infrared perspective. So it's a cool idea, yeah. All right, over here.
All right, let's talk about pregnant does.
Ooh, okay.
All right, and if you find a pregnant doe in your study, was the fawn or fetus also infected?
That is an excellent question. So the question was about pregnant does. If you find a pregnant doe, is the fetus also infected? So that can be called vertical transmission, where it goes from mom to fetus or baby. And so our lab has not been doing work on that, but there are folks who have been doing work on that. There's some evidence to suggest that it can be vertically transmitted, that it can transmit to fetuses. I would say, this is my disease ecology. Let's look at the big picture question. Baby born to an infected mom is going to have a heck of a lot of direct interaction with mom over the first chunk of life. So whether or not they got it in utero or they get it later, if mom's got it, it makes sense that baby's going to get it, right? That's my kind of disease ecologist big picture response. Yeah, Laura.
I'm just curious, are infection rates the same between does and bucks? I saw a slide that seemed to indicate that bucks between CWD and hunting have a much shorter life expectancy.
Sure. So the question was about infectious rates or infection rates between bucks versus does, and also kind of linking to survival in bucks versus does. So typically, when CWD enters a new area, you tend to see higher prevalences among males than females. There's a lot of hypotheses about why that might be. A big one is that bucks just have a ton of direct interaction with other individuals during the breeding season. So that's kind of generally the leading hypothesis or expectation for why we might see that. But certainly in our area, we see plenty of infection within does, but we also have a really high prevalence at this point. And so I think it's kind of reasonable to expect that does would have kind of caught up. In terms of survival, we do tend to see shorter lifespans for bucks in our area because they are under a lot of harvest pressure. So if you look at, kind of the age distribution for males versus females, we have a lot more older females than we do older males. We've also seen that among our animals who died in end-stage disease, or among our animals with CWD, our females were more likely to die in end-stage disease, which suggests to me that the males tend to get removed from the landscape earlier when they're in earlier stages of infection because they have that much higher hunting pressure. So males just generally are going to have lower survival than females, with or without CWD, right, just from the hunting. But that also then means that our does are perhaps staying on the landscape longer with their disease and maybe doing more shedding. So it's complicated, of course. I'm a disease ecologist, of course it's complicated. And then there's a lot of questions about, like, do does or do bucks, you know, how is their survival further affected by CWD? I don't know that we know that per se, but I just think they really are getting removed from the landscape earlier. So, yeah, over here.
There must have been thousands of CWD positive deer consumed by Wisconsin sportsmen in the last 30 years. How many people have actually gotten CWD?
Great question. So the question for the recording is, there have been, probably thousands of CWD infected deer consumed by Wisconsin hunters. Has anyone gotten CWD? The answer is, as far as we can tell, nope. There have been lots of long-term studies looking for weird prion neurologic diseases in humans. In humans, kind of our main prion disease that we think of is Creutzfeldt-Jakob, and with mad cow disease, we saw variant Creutzfeldt-Jakob, where people who ate infected cattle were developing this kind of other form of Creutzfeldt-Jakob disease. We haven't seen something like that in association with CWD.
At what point do we say, okay, it's safe to eat deer, but we'll work on the deer problem itself and worry about deer and quit scaring the crap out of people?
So the question for the recording is, when are we going to stop scaring people? So I think from a public health perspective, so remember, I'm also a veterinarian, and I'm never going to want to be the person to say, yeah, go ahead, eat that diseased animal. I don't think anybody really wants to be somebody to say that, because there's always that question of, we don't know. A rare event, even something incredibly rare, if you've got 1 million deer eaten, can suddenly stop being a rare event, right? Or it starts to, something happens, and it can start to, like, it occurring ever is kind of a concern for folks, right? I think a reasonable concern for people. There's also a concern that if CWD were to, say, go into things like swine, that the prion can change a little bit and then be able to better go from swine into people. So I don't know that we're ever gonna be able to say it's totally safe, right? You can never say that anything is totally safe.
You can't say that about anything.
Exactly. So I wouldn't say to eat it. I really wouldn't say to eat it, and I'm never gonna be comfortable saying you can eat it, because it's just too big of a what if.
We get on airplanes and cars, and the chances of death are much higher by getting on an airplane than eating.
Smoke a cigarette, drive on the beltline with your cell phone, have sex.
But there are some things that we can control, like whether or not you choose to eat a CWD infected deer, right? I can't control.
Boxes.
So, anyway, so
Last question.
you had your hand up earlier, so.
Based on my limited knowledge, it's unlikely that CWD occurred spontaneously in the wild herd. It's probably very likely that it came from licensed game farm cervids. If that's true, is there any way that the caged cervids could be made immune to cut the transmission from one to the other?
Sure, so the question was about kind of a little bit of the origins of CWD and this expectation that it couldn't have arisen sort of naturally within a wild deer herd, and that it maybe came from captive cervids into wild cervids. Now I'm being recorded, so no. So, and then of course, if captive cervids could have some sort of immunity that could then kind of cut off transmission between the two. So I will say where CWD came from is also hotly debated. One of the leading hypotheses is actually that it did arise naturally in herds, but has then moved to different areas, not always through natural movement of animals, right? So it might be movement of deer, whether by hunters or captive cervid producers moving from, that's how it can maybe make these longer distance movements. But there has been CWD that's shown up in Scandinavia, and that appears to be a totally different strain that arose naturally in Scandinavia. So we do have reason to believe that it can pop up on its own. That's how a lot of Creutzfeldt-Jakob, that's typically how Creutzfeldt-Jakob shows up in humans. It's just spontaneous and shows up in people. In terms of that interface between captive and wild cervids, so again, CWD isn't something that we can really vaccinate for at this time, and so kind of a herd immunity thing isn't really in the cards. There's a lot of work going on looking at, can we select for more disease-resistant deer? So that's kind of how they've tried to manage scrapie, or have been more successful in managing scrapie, is to selectively breed for sheep that are more resistant to that disease. However, what I didn't necessarily talk about, I kind of inkled to. So just because a deer is resistant to CWD doesn't mean they never develop it. They might still get the disease, and they might just live longer with their disease, which gives them more opportunity actually to transmit, right? So we have to be careful with, hoping that there's going to be a silver bullet that can help us just completely eliminate CWD, like selective breeding or something like that. So people try to use that as a reason to release deer onto the landscape that are like resistant to CWD and trying to do all sorts of meddlesome things with the deer herd. And so I try to, kind of be careful in talking about that, I guess. Yeah. Thank you all for having me. It was a real treat to be here. Thanks.